So we're going to spend the next 40 minutes or so talking about methyl donors from transition into lactation. And methyl group or methyl donor is basically chemically an alkyl derivative, which is basically a carbon that's not completely occupied, all four valences with hydrogen. So it would be basically CH3. And we like to think of carbon as an energy source, but in this particular case, methyl groups can have numerous cellular functions, such as methylation of genes, DNA, what we call DNA methylation. They play a role in phospholipid synthesis, and they play a role in protein synthesis because they can affect the transcription of genes and therefore the message that's used for synthesizing amino acids. And they have a dietary origin, and they come from three main molecules. Most likely methionine, quantitatively, it's probably the major methyl donor. But there's also folic acid or folate, an intermediate of folate metabolism can donate methyl groups. And another one would be an intermediate of choline metabolism. So choline, once undergoes oxidation, can produce betaine, and betaine is one of the methyl donors present in cells. So we have a multitude of compounds of different origins that can be methyl groups, can provide methyl groups through transmethylation reaction. So one of the effects of methylation is that it can affect genes. It can transfer methyl groups to the DNA. And the site of methylation is usually a cytosine, which once undergoes methylation, it becomes 5-methylcytosine. And this reaction is carried out by an enzyme that's called DNMT, or DNA methyltransferase. And it requires a donor, which can be one of those compounds that I mentioned before. In this particular example here, I'm using S-adenosylmethionine as the methyl group donor, which you can see it's being transferred to cytosine to form 5-methylcytosine. So why is this important? It's because during this process of methylation of DNA, that alters how genes behave, or how the chromatin behaves. So here I have a diagram of a piece of chromatin that contains multiple genes in the chromosome of a cell. And if this chromatin is loose and usually not methylated, it can undergo transcription to generate the message for subsequent protein synthesis. But if it turns out that some of those genes become methylated, such as in the example that I provided before, the addition of methyl groups prevent normal gene transcription. And as a general rule, it's not always the case, but as a general rule, methylation works by silencing genes because it attracts proteins such as histone deacetylase or methyl-binding proteins, as well as other co-repressor proteins that blocks transcription factors to bind to the promoter region of the genes, and therefore mRNA synthesis is suppressed. So at the end of the day, methylation reactions can control to some extent protein synthesis by controlling at the level of transcription, and this leads to what we call gene silencing. Okay? So that's one aspect of one-carbon metabolism. Another aspect of one-carbon metabolism is that methyl donors play a role in phospholipid synthesis, and we know that phospholipids play a multitude of roles in cell biology. And phospholipid can be synthesized de novo through this pathway that's called CDP-choline pathway, in which choline of dietary origin can be activated to produce phosphocholine by the addition of a phosphate group from ATP. And this can undergo reactions to form CDP choline that then condenses with a set of fatty acids, usually a group of long-chain fatty acids in diacylglycerol to form phosphatidylcholine, which is the major phospholipid in most mammalian species. There is a second pathway that's typical of the liver that's called the salvage pathway. And as the name says, it is used to recover substrate or molecules that are part of the phospholipid group. So in this particular case, phosphatidylethylamine can undergo a series of methylation reactions coming from S-adenosyl methionine to produce phosphatidylcholine. And phosphatidylcholine is one of the phospholipids that make up what we call lecithin. And lecithin, or phospholipids in general, they are building blocks for cell membrane, but they have this feature of being amphiphilic with amphipatic properties. So they have a polar head that is usually hydrophilic, and they have a tail that's usually lipophilic. And that becomes very important when you're transporting lipid-like or fat-like compounds in aqueous solution. So for example, here I illustrate a molecule or a compound called very low density lipoprotein that is secreted by hepatocytes to carry triacylglycerol, cholesterol esters from the liver into the general circulation to redistribute lipids to other tissues. Obviously, those lipids would not easily mix in blood unless they are emulsified. And one way of combining these lipids in a compound that can be transported in blood is by incorporating proteins or phospholipids into this big compound. And here I illustrate the location where phosphatidylcholine or this lipotropic compounds such as phosphatidylcholine, phosphatidylethylamine can be present that combines this polar head with the lipophilic tail. So this becomes really important for fatty acid transport, not only in the blood as the liver secretes lipoproteins, but also absorption from the gastrointestinal tract, as well as transport across the epithelial cell in the mammary gland. So it has multiple roles there. And there are multiple pathways that are interrelated. And I'm going to give you one example here of those pathways using a central molecule that's called choline, but I could pick something else. So in this particular case here, choline, which is a water-soluble molecule, can undergo acetylation to form acetylcholine in neurons or in the nerve tissue, which is an important neurotransmitter in the central and peripheral nerve system for communication between neurons. Or you can undergo oxidation to form betaine, which is one of those methyl donors. As you can see, it contains 3 methyl groups. And when betaine donates a methyl group to homocysteine, it can form dimethylglycine, which now has only 2 methyl groups, and one of those methyl groups is transferred to homocysteine. Homocysteine can be used to resynthesize methionine through an enzyme called methionine synthase, in which homocysteine receives a methyl group from 5-methyl tetrahydrofolate. And just to be clear, this morning there was some discussion. I just want to point out that this enzyme here, methionine synthase, requires a cofactor, which is cyanocobalamin, which is vitamin B12. So this is an enzyme that for proper function requires B vitamins, and many, if not all of the B vitamins work one way or another as a cofactor in those biochemical reactions. So to no surprise, in this particular example, cyanocobalamin is a cofactor in this reaction that facilitates the transfer of a methyl group from 5-methyl tetrahydrofolate to methionine, resynthesizing tetrahydrofolate. So here we see an interplay between compounds that originate eventually phospholipids, B vitamins, and amino acids. Methionine can undergo further conversion and accept an adenosyl group and form S-adenosyl methionine, which is another methyl donor that can donate a methyl group per step to synthesize phosphatidylcholine in what's called the salvage pathway, as you will see. So I showed you before that choline can be synthesized through the de novo pathway that's called CDP choline pathway, which is this here, or choline can be synthesized in the hepatic tissue through the salvage pathway that requires phosphatidylethylamine that receives methyl groups from S-adenosyl methionine to be converted eventually in phosphatidylcholine. So here we see the connection between or among B vitamins, amino acids, and phospholipids becoming very important. So that's another role of one-carbon metabolism that becomes really important. There's another one that's called the transsulfuration pathway that becomes really important in terms of providing antioxidant molecules. That's not illustrated in this diagram here, just for the sake of simplicity. Otherwise, we would spend a lot of time just talking about those things. So why is this important for the transition cow? Well, we know, and you know very well, that transition cows have reduced intake as they approach parturition, particularly in the last week of gestation, and they have inadequate intake of dry matter in the first weeks after calving, which lead to negative nutrient balance. Because of the diets that we feed prepartum that typically contain a large amount of NDF, and in many cases very undigestible NDF, and because intake is low, it's no surprise that there may be a possibility for inadequate intake of essential amino acids because microbial synthesis is reduced because there's just less substrate in the rumen for microbial growth. And methionine can be decreased in supply, and we know that methionine is typically one of the limiting amino acids for milk protein synthesis in typical diets based on corn and soybean. But also because the synthesis of phospholipids require transmethylation reaction, and a feature in the ruminant is that when cows or calves or heifers consume dietary ingredients that contain choline, it turns out that the rumen microbes degrade most of that choline in the rumen. Therefore, very little reaches the small intestine to be absorbed. So the pathway that I showed you initially, very likely the de novo synthesis of phosphatidylcholine from dietary choline most likely is in short supply during this period because degradation of choline in the rumen and the low dry matter intake. On top of that, we know that amino acids are limiting because right at this time there is synthesis of protein in colostrum that's sucking up a lot of the amino acids that have been absorbed. So very likely the supply of methionine for transmethylation reaction through S-adenosyl methionine may also be in short supply. So we would expect that availability of phospholipids in this period would be lowest compared to other stages in the lactation cycle, and I'll show you some data on that. Well, this is important because in the many aspects of intermediary metabolism in transition dairy cows, lipids or fatty acids become a central point, and we know that during this period in which the mammary gland sets a priority for nutrient utilization, cows rely on adaptation to increase the rate of lipolysis of fat deposits that are needed to supply calories for the mammary gland. So there will be mobilization of triacylglycerol that results in circulating free fatty acids that are transported in blood associated with albumin primarily that can be transferred. Well, I don't know why my computer connect with this. Let me close this. That can be transferred to the mammary gland and provide mobilized fatty acids, mostly long-chain fatty acids, C16 or longer, for milk fat synthesis. But some of those fatty acids can also be taken up by hepatocytes through a transport system and they will be a source of energy to the hepatic cells. And during this process, not only will generate ATP, but some of them can undergo ketogenesis once the energy needs of that cell is met. So now the cow generates ketone bodies that can carry carbon sources in a water-soluble form that can be used by other tissues, such as the muscle, the brain, or even as a substrate for synthesis of milk fat through the de novo pathway of milk fat synthesis. But if the amount of fatty acids taken up overcome the ability to synthesize ATP, synthesize ketones, and export those fatty acids, they eventually accumulate as triacylglycerol because those fatty acids will be re-esterified within the cytoplasm of the hepatocyte. These triacylglycerols can eventually be exported from the liver as very low density lipoprotein, but it turns out that ruminants have limited ability compared to monogastrics to secrete VLDL. And as those triacylglycerols accumulate in the cytoplasm of the cell, it can induce inflammatory response, it can induce acute phase response, and it can damage the cell and alter its metabolic function. So we don't want excessive amounts of triacylglycerols accumulating in the hepatocytes of dairy cows, although most of them will have some degree of accumulation. So are there nutritional interventions that can play a role in these processes that would benefit cows? And we're going to spend some time talking about them. So first I want to touch on how common is fat accumulation in the hepatic tissue in dairy cows. And there is a really extensive review in literature published about 15 years ago in which they summarized multiple studies in the literature that had collected hepatic tissue. And what they showed was that if we use the cutoff of 5% triacylglycerol in the wet tissue bases of the liver, many cows will have that amount. So typically in a well-fed cow, they have 1, perhaps 2% triacylglycerol in the hepatic tissue. But as you can see here, anywhere from more than 15% of the cows to as much as 65% of the cows had between 5 to 10% triacylglycerol, and another fraction of the cows had more than 10% triacylglycerol. So if we put it all together, what we see is that almost half of the cows will develop some degree of moderate fatty liver during the first weeks after calving. So usually that accumulation is maximum between the first and second week, and then slowly those triacylglycerols get eliminated or they get oxidized from the liver. So are there things that we can do to reduce this? Okay. So obviously phospholipids have lipotropic effects. And by having these lipotropic effects, they play a role in transfer of triacylglycerols from the hepatic tissue among other tissues. So here you can see in a case control type of study in which this group, they looked at the amount of microsomal transfer protein, the abundance of a apolipoprotein B100 and a apolipoprotein E. Those are all apoproteins that are important for VLDL assembly and secretion. And this is a transfer protein that transfer triacylglycerols across cell membranes. And what they showed was that cows that developed fatty liver, they had smaller abundance of those important proteins for fatty acid or triacylglycerol metabolism within the hepatic tissue. In monogastrics, in mice, supplementing choline to animals that were fed a choline deficient diet, as you can see, enhanced the ability of the hepatic, of the intestinal cell, the enterocyte, to secrete apolipoprotein B48. And this is the main apoprotein that's incorporated into chylomicrons during the absorption of fatty acids in the lymphatic system. So this would make the case that synthesis of apoproteins or VLDL assembly can be compromised in cows that develop fatty liver, and maybe there is some relationship in terms of cause and effect there. And this also shows that fat absorption in the gastrointestinal tract can be compromised when the availability of substrate for phospholipid synthesis is less than ideal. Okay. So as I mentioned to you, it's no surprise that because of what happens during the transition period, it's right at the time when the availability of phosphatidylcholine is least in dairy cows, right between the week of calving and the first 1 or 2 weeks after calving. So we would think that, hey, if I can enhance the availability of phosphatidylcholine one way or the other, perhaps this would benefit transition cows, and we'll touch into that during this presentation. Now, we know that phosphatidylcholine has lipotropic effects, and we know that if we deliver substrate for its synthesis, in this particular case, by supplying choline in a rumen-protected form might be beneficial. So this is what these two experiments showed. In the first one, they used a dry cow feed restriction model, and they looked at cows that received or did not receive choline in a rumen-protected form. And then they measure triacylglycerol in the liver during the feed available time, and then after cows underwent feed restriction. And what they show was that by causing cows to not consume feed, they mobilize body fat that accumulates in the liver, but the accumulation is less when they receive supplemental rumen-protected choline. The same was observed in this experiment by Marco Zenobi here in Florida, but now he titrated the dose of choline ion in a rumen-protected form. He fed from 0 grams of choline ion all the way up to almost 26 grams of choline ion. So this would be approximately 30 or 32 grams of choline chloride being fed in a rumen-protected form. And what Marcus showed was that during feed restriction, these cows had a linear decrease in hepatic triacylglycerol accumulation in the liver as the dose of choline increased. We just replicated this experiment recently with exactly 110 pairs prepartum cows. These cows were at 232 days of gestation, and they were assigned randomly to one of 5 treatments. So we had 22 cows per treatment, and I'll go through the basics of the experimental design. So we had a control group in which cows received no choline ion. And then we had a treatment in which cows received 12.9 grams of choline ion in a form that has low concentration of choline chloride in the entire product. So there's a lot of choline lipids. Or they receive almost 26 grams of choline ion in the same form. The next 2 treatments, we use exactly the same dose, either 12.9 or 25.8, but now they were supplied in a rumen-protected form with a high concentration of choline chloride. The experimental design was quite simple. Cows underwent 5 days of ad libitum intake, and then we imposed feed restriction for 9 days, in which they were allowed to consume 50% of their net energy for lactation required for maintenance and pregnancy, assuming a 42-kilogram newborn calf. So I'm showing you some of the results. So as expected, we controlled intake during feed restriction. These cows were consuming about 7 megacalories of net energy for lactation, and this is exactly 50% of the estimated amounts of net energy required for those cows based on NRC. Okay. So to no surprise, concentrations of NIFA and BHB, they were elevated for a dry cow, but they were exactly the same across the different treatments. So we were interested whether the amount of choline or the form in which the rumen-protected choline was supplied would affect hepatic triacylglycerol accumulation. And as we had seen before in previous experiment, we had exactly the same response. So when we look at hepatic triacylglycerol after 9 days of feed restriction, on a as-is, on a tissue basis. In the control group, we had more than in the choline-treated cows. And as we increase the dose of choline, we can see a further reduction. So we had an effect of choline supplementation and amount of choline supplementation. The exact same response was observed when we cracked the triacylglycerol to tissue dry matter basis. When we look at glycogen content, it increased when cows received rumen-protected choline compared to the control, and the same was seen on a dry matter basis. So when we look at the ratio of triacylglycerol to glycogen, it was more favorable in the cows that receive rumen-protected choline, in particular those that receive a larger amount of rumen-protected choline. So we can see that in the cow, as well as in the monogastric, in which the literature is much more abundant, choline in fact has this lipotropic effect. We just don't know the exact mechanism yet, and we are working on that as well as other people are. Now let's just look at what happens when we feed this through the transition period. Here I have 3 experiments that show different responses. In the first one, the concentration of triacylglycerol in the hepatic tissue was less for the choline supplemented cows. In the second one, there was no difference. In the third one, choline supplemented cows actually had greater concentration of triacylglycerol. So this heterogeneity in the data, in general, is associated whether choline stimulated or did not stimulate dry matter intake, because in almost every experiment, choline has been able to stimulate milk yield, in particular fat-correct milk yield. So when milk yield increase without a concurrent change in dry matter intake, we see less of a positive effect or no positive effect on hepatic triacylglycerol. But when intake is stimulated, we see a marked reduction in triacylglycerol in the liver. We recently reviewed literature to look at responses to choline supplementation starting prepartum and carry on through the early postpartum period. And during this process, you go through searching the literature, in which we found lots of papers that cite the word choline, but many of them were either duplicated in different engines that we use to search. Eventually, we screened records. Many of them had data that we were not interested. So we were interested in choline for prepartum cows fed in a rumen-protected form. So we were able to find 20 papers with 21 randomized experiments in which choline was randomly assigned to cows, yes or no. And we were interested in looking at animal responses such as production responses, as well as health responses. Just for the sake of time, I won't be able to show you everything, but I'll touch in some key aspects. We were interested in answering the question, what is the dose of choline that optimizes animal response, and whether there was an interaction between choline and supply of methionine in the pre or postpartum diets of those cows. And I'll show you data on milk yield, energy correct milk yield, and milk components. So here's just to show there were 30 treatment means in which cows received no choline. In every single experiment, there was an untreated control group in which they receive 0 grams of choline ion. And then there were 36 treatment means in which cows received some amount of choline that ranged from 5.6 to 25.2 grams. So this is the range of choline intake in the cows, given that these cows were supplemented with choline. But there was always a control group within the same experiment. And you can see here what was the intake of methionine in those cows prepartum and postpartum. And I'll show you a histogram here for the cows that were in those experiments during the prepartum period. So you can see intakes of metabolized methionine that average approximately 25 grams, and that represent approximately 2 to 2.1% of the metabolizable protein. And there were some experiments that supplemented protected amino acids to a very large amount, so they got to values that were quite high. If we look at the postpartum diets, we see larger quantities because these cows were eating more dry matter. But if you look at the ratio of metabolized methionine to the metabolizable protein of the diet, it follows somewhat of a similar pattern with a few experiments in which postpartum diets were supplemented with a substantial amount of rumen-protected methionine. So let's just look at what happened to intake. You will see figures like this. The dots that fall on the 0 grams of choline ion are the control treatment means, or the 0 grams. In this Y scale, you're going to see the response. In the X-axis, you're going to see the amount of choline ion in those mixed models. The size of the symbol represents the weight of that treatment mean in the statistical analysis. We use meta-analytical approach to this. The lines connect treatment means within the same experiment, and the continuous dark line represents that there wasn't a statistical effect. And here is the direction of the response, which you can see it's a linear response with an increase of this level for each gram of choline that was added to the diet during the transition period. If we use the median amount of choline ion fed in those experiments of 12.9 grams, we observe a 230 gram increase in dry matter intake prepartum, a 0.5 kilogram increase in dry matter intake postpartum. So that's dry matter intake. There's a slight increase. It's not a huge increase. So here's yields of milk and energy correct milk. We see a linear response to choline supplementation in terms of milk yield and in terms of energy corrected milk yield. Okay, but pay attention to this. There is an interaction between choline and the proportion of methionine as the metabolizable protein in the diet. So this interaction here means that the response to choline was to some extent dependent upon the supply of methionine. So here we see that pathway that we just discussed in biochemistry playing a role in cow biology and animal response. So here is a forest plot that shows the energy corrected milk across the multiple comparisons. So you can see what direction that response went. So if the point estimate, which are the black dots, fall to the right of this central line, it means that supplementing choline increased milk yield or energy corrected milk yield. If the point estimate falls to the left, it means that it numerically decreased milk yield. Okay, you can see the confidence limits in the overall response, which is the dashed red line with the random effects here showing that there was an increment in energy correct milk across all comparisons of almost 0.4 standard deviations, which translates into approximately 2.2 kilograms of milk. But I mentioned to you that this was dependent upon supply of metabolizable methionine, because we observed some heterogeneity in the response because the magnitude of response was dependent on supply of methionine. And this is how the data looked like. When diets had moderate to low proportion of the metabolizable protein as metabolizable methionine, the response to choline was substantial. Okay, but when diets had a quite large amount of the metabolizable protein as metabolizable methionine, now the response to choline was still present but became smaller. Okay, and the same was true for energy corrected milk. So if we look at when metabolizable methionine was 1.8% of the diet, of the metabolizable protein of the diet, the response to 12.9 grams of choline ion was 2.1 kilograms of energy corrected milk. When cows received the same amount of choline ion in a room protected form, but the diet now contains 2.3% metabolizable protein, they still respond positively, but the response was less, now 1.2 kilograms of energy corrected milk. So here we can see that there is a relationship between these molecules, but there is also some independent effects because we see responses regardless of the level of metabolizable methionine, but they attenuate at the very high amounts. So here's concentration and yield of milk fat. Milk fat goes up. There was a quadratic relationship with choline, with the optimum amount somewhere here in terms of concentration of fat, but the yield respond exactly like energy correct milk or milk yield. The same was true for milk protein. We saw a linear response to amount of choline supplemented to cows. So this is telling me that choline is able to stimulate milk production. The interesting thing, and I'll touch on a couple of other aspects about this source of methyl groups, is that the response goes above and beyond the period of supplementation. This is one of two experiments that we conducted here at the University of Florida in which cows were supplemented during the transition period, starting 3 weeks before the expected day of calving up to 21 days postpartum. So on average, these cows stayed in prepartum diet about 18 to 19 days and until exactly 21 days postpartum. And you can see that there was an increment in milk yield, but this increment in milk yield lasts for quite a long period of time. In fact, for the first 40 weeks postpartum, there was a tendency for an increase in milk yield of about 2 kilograms in this particular experiment. In the second experiment, in which cows were supplemented again during the same period of time in their lactation cycle, we saw an additional 2 kilograms milk response up to 25 weeks postpartum. So, to no surprise, things that we do during the transition period that benefits cows, they tend to last longer than the period of supplementation because many of those interventions have impacts on animal health and they have effects that are other than just the nutritional component of the particular supplement that you are providing. One of the pressing questions that many people ask is, what cows respond to choline? Are only the ones that are more prone to fatty liver? So we pulled together data from 2 experiments in which we collect body condition score twice before enrollment in the experiment. And we had cows that received no choline supplementation, or they received 12.9 grams of choline ion in a rumen protected form. Okay, so you can see here the distribution of body condition score of those cows, and you can see that they are quite similar across the 2 treatments. So we had cows that had moderate body condition score, and we had cows that were overconditioned. So we would perhaps would expect that these cows would be more likely to respond to choline because of the notion that choline has these lipotropic effects, and overconditioned cows usually are more prone to disease that are associated with intermediary metabolismal lipids. But what we found was that the response to choline, which is the difference between the blue line, which is the control, and the orange line, which is the choline supplemented cows, for milk yield and for energy correct milk yield, as well as for every response that we measure, was consistent across the multitude of body condition scores. So each dot here represents a cow in the data set. There's about 215 cows here. And the point I want to make is that you can see the response is an increment in milk yield, an increment in energy corrected milk yield of approximately 2.2 kilograms, irrespective of body condition score. We saw no interaction with body condition score. So my point here is that cows respond to rumen protected choline whether they have low or they have high body condition. So because choline is interrelated with supply of metabolizable amino acids, in particular methionine, I'm going to finish up just showing you some data of protein in prepartum cows. We did the exact same exercise as I had shown you before for collecting data in the literature from published randomized experiments in which the cows were supplemented or not with choline. We did the same with protein prepartum. But now these experiments had to feed different diets prepartum and manipulate only protein, either the amount of protein in the diet, the degradability of the protein source, or supplement amino acids, but only prepartum. The postpartum diets, they were always the same. Okay, so at the end of the day, we were able to identify 27 experiments in which cows were randomly assigned to receive different concentrations of protein in the diet or different protein sources, or be supplemented with amino acids prepartum. And I want to emphasize that because you are going to see subsequent webinars in which people are going to focus on supplementing amino acids pre and postpartum in a lot more detail than tonight. So we were interested in understanding whether protein prepartum affects postpartum intake, yields of milk, fat-correct milk, and milk components, body weight, and body condition score. So what we did is we pulled the data from literature. We identified the diets and the dietary ingredients. We entered those diets into NRC. We entered the observed dry matter intake of the particular treatment and the treatment mean. And then we calculated the supply of net energy, metabolizable protein, and metabolizable amino acids. And we analyzed the data for nulliparous separate from parous cows, just because, as you will see, nulliparous eat less. So the supply of metabolized protein is quite different. So the dots here represent the diets that were fed in the different treatments to nulliparous, which are the triangles, and to the parous cows, which are the open circles. And you can see that for a given amount of crude protein in the diet, there's a substantial difference in the supply of metabolizable protein. And the reason is quite simple. Cows eat more than heifers, and by simply eating more, there will be more microbial growth. There will be more undegradable protein that gets to the small intestine. So the supply of metabolizable amino acids goes up just because of the differences in intake. So if you look at two particular amino acids, metabolizable methionine and lysine, we see exact same response. Parous cows consume more of those. In some diets, cows were supplemented with protected amino acids, so then the amounts were substantially greater. So that is true for methionine, lysine, or any amino acid that we looked at in that data set. So just quickly, I'm going to show you the data on heifers, or first lactation animals. They were nulliparous cows before calving. Now they became first lactation. And in parous cows, the same system. The size of the symbol represents the weight of that particular treatment mean in the analysis. If you see a central dark line, means that there was a statistical effect, which I didn't place here. But if that central line is not present, there was no statistical effect, okay? And the dashed lines connect the treatment means within an experiment. So to no surprise, as the intake of metabolizable protein prepartum increased, and our upper limit observed was 1,100 grams for the heifers, there was a linear increase in milk yield, in energy-corrected milk yield. But when we looked to the data from the parous cows, we saw basically no response. Whether the cows ingested 900 grams of metabolizable protein, or they consumed 1.4 kilograms of metabolizable protein prepartum, we saw exactly the same milk response or energy-correct milk response. So no benefit to parous cows, a major benefit to nulliparous cows. When we look at fat yield, exactly the same as I showed you before. Protein yield exactly the same. So major benefit to nulliparous cows, and there are reasons for that. Not only they have a growing fetus, they are creating lean tissue, so they need to add muscle mass during this period. Whereas if we look at the parous cows, we saw no response in fat yield and a small response in protein yield that was only observed in the experiments that had cows producing more than 36 kilograms of milk per day postpartum. In experiments that had cows producing less than 28 kilograms of milk per day, we saw no response to supply of metabolized protein prepartum. So if a cow consumed 1,100 grams of metabolizable protein prepartum, the estimated supply of metabolizable lysine was approximately 92 grams or 94 grams, and the supply of metabolizable protein was approximately 26 to 28 grams. Okay, so if we want to translate that into individual amino acids. Okay, just to summarize and conclude, supplementing choline ion as a rumen-protected choline to feed restricted cows it's unquestionably that it has major lipotropic effects, and it reduces triacylglycerol and the risk of fatty liver. Supplementing choline to transition cows benefit lactation performance, and this benefit goes beyond the period of supplementation. We saw responses observed in diets with low or high metabolizable methionine postpartum, but there was an interaction suggesting that there is an interplay between them, but there's also some independent mechanism present there. And the response were observed regardless of body condition score of cows. We really don't know the optimum amount of choline ion. We think that's probably closer to this 25 grams, but we need more data on that. Just lastly, the amounts of metabolizable protein prepartum, they differ between the nulliparous and parous cows, and I think there's good biology to justify that. Based on the published data, we think that parous cows need anywhere between 800 to 900 grams of MP prepartum, which approximates to 20 grams of metabolized methionine, whereas heifers, nulliparous, need at least 1,100 grams. We don't know whether they need more because there are no data on that. Okay, and this results in approximately 26 to 28 grams of metabolized methionine prepartum for heifers. So thank you for your attention, and I'll be glad to take any questions.